Alrighty, hello y'all. It's Reese here, and it is time for discussion number 4. You just had your first exam. Hopefully it went well. And now we are hopping into some new material. So I'm going to go ahead and share my screen and turn my camera off because I forgot to do that last time, and you could just saw like my chin the whole time. So I'm going to turn my camera off. I'm going to share my screen, and we will go ahead and begin. Real quick before we hop on into this, I did want to point something out. I got a few emails asking about where the discussion videos are posted. So where you can find them is under the announcements tab. You'll see TA discussion recordings. Click that, and each time I record a recording, I'll go put it on YouTube and upload this list with that. I just don't want to send out a different announcement every single time. I think that would fill up your inbox with unnecessary announcements. So this is where you can locate those. Alrighty, so hopping on into this. So today we're looking at neurophysiology and the central nervous system. So the first thing we're going to do is a little bit of anatomy. We're going to do some anatomy review, looking at the structures or the anatomy of a neuron. So hopefully a lot of this is kind of memory for you from anatomy classes if you have ever taken any. But what we have for A is the cell body or the soma. So A is our cell body or our soma, kind of like the integration center. B is the dendrites. So they're like the arms of the cell that will grab and receive signals. C is going to be the axon hillock, and that will have to do with threshold, and we'll learn about that in just a little bit. D is the actual axon itself that will send signals. E are little structures called myelin that will form the myelin sheath, kind of blocking off space on the neuron membrane. There are little spaces in between each of those myelin filaments that are called the nodes of Ranvier, and we will learn, or Ranvier, and we'll learn about those in a little bit as well. And then lastly, F is our axon terminal, where those impulses or signals that were being sent down, the axon will actually leave the axon. So that was a little bit of A and P review by looking at those structures. So now we're going to kind of get into actual physiology. So before we move on to B, which is our first physiology question, I wanted to review our 3 types of channels, because I think with this, it makes B a lot easier to answer. So the first channel that we're going to look at are the ligand-gated channels. So we call these ligand-gated channels. And what ligand means is that there is some type of receptor on the cell membrane, the ligand. And oops, I just realized something. Let me do my small eraser. These should be open because that's what we're going to be showing, is those opening. And if you wanted to pause the video and draw this out, that could be helpful. So we have our ligand-gated channels. And so what happens is we, for example, will have some type of ion. We can use sodium because we use that quite often in this class. And what we see is that sodium can't just move into the cell. It needs some type of channel, and for sodium, moving into the cell, in this case, we'll use a ligand-gated channel. So what we see is that there will be some type of neurotransmitter floating around. And that neurotransmitter is going to bind to that ligand. And when that happens, when that neurotransmitter binds to the ligand, that sodium will now be able to move into the cell, past that cell membrane. So that's kind of how a ligand channel works. Sodium is outside of the cell, needing to move past that cell membrane. However, it can't just do that. So we have that specific ligand-gated sodium channel. Some neurotransmitter will bind to it. So we'll have a neurotransmitter bind to that channel or that receptor, that ligand receptor, and that will allow sodium to move into the cell. So that's our first type of channel. Our second channel is called a voltage-gated channel. Voltage-gated channel. And what we see is that it works a little bit differently. So a cell at rest is at -70 millivolts. Voltage. We're looking at voltage with voltage-gated channels. And if we have some sodium that needs to move in, again, it can't. But a way we can do this is by using a voltage-gated channel. So something will happen inside the cell that will cause it to change voltage. And what we see is that this change in voltage is going to allow that channel to open. So we have a voltage change, which is going to open the channel. So now that sodium is able to move into the cell. Now, when that sodium moves into the cell, we're going to have another voltage change, and we'll look at this more in depth in just a little bit. But sodium is going to cause the cell to become more positive. So for example, it could depolarize to like positive 30 millivolts, and we'll look at that more specifically in a little bit. So our last type of channel that we have is a mechano-gated channel. So a mechano-gated channel. And we'll be learning about these in the future, but I'll just give you a little overview, but we're not going to go over these too in depth. But what we see is that if we had like a sodium ion that needed to move into the cell and couldn't, we could also use mechanical functionality to do so. And so we would see these types of channels in like skeletal tissue. And what would happen is we would actually have like a stretch, and that stretch would open up that channel, that physical stretch. And that's what would allow that sodium ion to move into the cell. Again, we're not going to hit those too hard right now, the mechano-gated channels. These first two are what are more important. So real quick, I just wanted to point out that these ligand-gated channels, we're normally going to find those at the dendrites and cell body. So we normally see the ligated channels at the dendrites and the cell bodies, or those somas. For the voltage-gated channels, we're normally going to see those at the axons and axon terminals. All right, awesome. So with that, we can kind of answer some of these questions. So if we are looking at where we would see voltage-gated sodium channels, what we would see is that we're going to find those at the axon hillock and the axon. So we find those at the axon hillock and the axon. And this will make a little more sense in our next example, but let's get these down now. Voltage-gated potassium channels, we would find them in the same place because, again, that's where we find our voltage-gated channels. Our voltage-gated calcium channel, this one's going to be just a little bit different. It's going to be at the end of our axon, at that axon terminal, and we'll see that in our next scenario. The ligand-gated channels, we haven't really learned about these. We'll learn about them more in, sorry, I keep saying that, in this next example. But where we're going to see those are mainly at that dendrite, so B mainly, but also at that soma or cell body. Same with the potassium channel. And then lastly, the ligand-gated calcium channels. This is kind of a little trick. We will not see these anywhere, though we only see voltage-gated calcium channels. So there are no ligand-gated calcium channels, only voltage-gated calcium channels. And we'll see that in just a little bit. So I wanted to go back up here and kind of go over an action potential, what's going on with the neuron during that, the different locations, and these ligand and voltage changes. So let's make up a little example here. So let's say we have like a neuron right here, and leaving the axon terminal are going to be a bunch of neurotransmitters. A bunch of neurotransmitters are going to leave the previous neuron and surround the dendrite and cell bodies. So what we see is that whenever those neurotransmitters arrive at those dendrites, we will have the opening of ligand-gated channels. Remember, we talked about how we find those on the dendrites mainly, and sometimes also on the cell bodies. So let's say we had some sodium floating around in our body, and we needed to get that into the cell. What we're going to use is that ligand-gated sodium channel. So ligand-gated ion channel would be for any ion, but here we're going to call it a ligand-gated sodium channel. And it will open when those neurotransmitters arrive. So it is opened, and now sodium can move into the cell, into the neuron. So what we see is that when sodium enters the neuron, it's going to become a lot more positive because sodium is positively charged. So we're going to have a change in voltage. And this change in voltage is going to cause the opening of some voltage-gated sodium channels. So we're going to have the opening of some voltage-gated sodium channels. I'll write that out for you. Voltage-gated, I can never say these, voltage-gated sodium channels. And those will open again because we have changed the voltage inside of our neuron, inside of our cell. So when those open, what we're going to see is that we'll have this type of saltatory conduction, which is this kind of like jumping motion from node to node. So those spaces in between those myelin filaments are called the nodes of Ranvier. Or Ranvier, I don't know how to pronounce it correctly, but those are kind of the little gaps in between the myelin. Myelin kind of blocks off the cell membrane. So instead of opening a bunch of different sodium channels along the axon, they're just going to hop from one node to the other. So this happens very quickly. So if we drew out a little chart, which I meant to do this a little earlier, this is kind of what would be going on so far. So we have a chart here. What we will label will be negative 90 millivolts, negative 70 millivolts, negative 55 millivolts, which if you remember, that is threshold. So that's what the cell has to reach, what the stimulus has to be strong enough to reach to cause an action potential. And when we have an action potential, we see that quick inflow of sodium, which will go up to about positive 30 to positive 40 millivolts. So what we started off with was that cell at rest. And then once we had that sodium, those ligand-gated sodium channels open, what happened was something like this. We kind of started to step up to threshold. And what we call these steps up is excitatory postsynaptic potential. The synapse is the space between the neuron and the dendrites. So where these neurotransmitters are existing are these synapse. So excitatory means we got more positive. Postsynaptic, once we entered into the cell, potential. So again, we see these with those ligand-gated sodium channels. So we hit that threshold, and then those voltage-gated sodium channels opened in blue there. And when those open, again, they will quickly travel down the axon. And it's really quick, looks something like that. And what we term that, I don't know if y'all remember this, what we term that is depolarization. Depolarization. And what depolarization is, in words, is that those voltage-gated sodium channels opening and sodium moves into the cell. So our cell becomes more positive. So what we see is that depolarization occurs really quick. And once that sodium moves down, we're going to have the opening of some voltage-gated potassium channels. And I will represent that here. I'm drawing little Ks. You can't really see them. They also would not exist on the myelin. They would exist in those nodes of Ranvier in between the myelin, but I don't really have space to draw that out. But when that sodium moves through, those voltage-gated potassium channels would open again because of that change in voltage from the sodium. So we can call these over here, those are the voltage-gated potassium channels that have just opened after that sodium moved through after that depolarization. So that looks something like this. So what is happening there is repolarization. So repolarization, and just for definition purposes, that is going to be the opening of that voltage-gated potassium channel. And when that opens, potassium will flow out or move out, making the cell become more negative again. It actually becomes hypernegative, and we call that hyperpolarization. And that lasts for a very short time. So those voltage-gated potassium channels open, and what we see next is we now move to that axon terminal. So we've gone through the dendrites and the cell body, and we are now at the axon terminal. So those sodium ions had moved down there, so we have this positive voltage down there, a change from what it was initially, and that sodium influx causes voltage-gated calcium channels to open. So that positive charge down in those axon terminals is going to cause voltage-gated calcium channels to open. So voltage-gated calcium channels to open. And that's again because of that change in voltage from that sodium moving down the axon. So when those open, what we see is that calcium, oops, I did not mean to do it in that color, change that real quick. What we see is that calcium can now move into the cell. And when calcium moves into the axon terminal, we see the initiation of exocytosis of neurotransmitters. So what that means is we will release neurotransmitters from that axon terminal to the dendrites of the next neuron. And the process will repeat. So what we had here was a cell at rest, and then some signal, some stimuli was caused by those neurotransmitters. They arrived at those dendrites. Once they arrived there, the ligand-gated sodium channels opened and sodium was able to move into the cell body. Once it moved into the cell body, the cell became more positive because sodium is positively charged. Therefore, we had a change in voltage. That change in voltage caused the opening of our voltage-gated sodium channel. So once those voltage-gated sodium channels opened, we had that quick conduction, that saltatory conduction of jumping from node to node very quickly down the axon. Once that sodium moved down, we had the opening of the voltage-gated potassium channels, which caused that outflow of potassium. And once we looked down there at the axon terminal, we saw that that positive voltage from the sodium caused us to open some voltage-gated calcium channels, which let calcium into the axon terminal, causing the initiation of exocytosis of our neurotransmitters. Those neurotransmitters were released from the axon terminal of that neuron to the next neuron until it reached its final destination. So that is kind of just a general overview of what's going on. And before we finish up, I wanted to hit on one more thing. So when we look here back at our graph, that hyperpolarization, again, is just when that potassium overflows just a little bit, but our cell will eventually move back to rest. But at that beginning part, we saw that EPSP, excitatory potential, or excitatory postsynaptic potential, but we also sometimes don't have stimuli that are strong enough. So sometimes it takes a couple different stimuli to get us to our graded potential to cause that action potential. So the EPSP is that upward trend, that more positive trend, and that more negative trend is called inhibitory postsynaptic potential. So for example, this would be like ligand-gated chloride channels opening and causing that cell to become more negative. So we have two different types of potentials. We have our action potentials and our graded potentials. And our graded potential has to be strong enough to reach an action potential. So everything you see above this line is an action potential, and it will only occur if we have a strong enough stimuli to hit threshold. And those stimuli, what we can think of them as is the summation or sum of our ESPS and our ISPS. So if that graded potential is strong enough, we will have a super threshold stimuli and we will have an action potential. However, if it's not strong enough, we will have a sub-threshold stimulus and we will not have that action potential. All right, okay, cool. So I should have done that example before B, but you can kind of look at these now again and see how those would make some more sense. So moving on to C, this kind of follows what we just looked at above. So what we see is that our EPSPs are excitatory postsynaptic potentials. Those are occurring in mainly the dendrites, but also on those cell bodies. And an example you can think of there is like those ligand-gated sodium channels opening, sodium flowing in, which makes the cell more positive, and we depolarize. So it's excitatory, it becomes more positive and leads to depolarization. Our inhibitory postsynaptic potential, same location, mainly at those dendrites, also at those cell bodies. And an example you can think of there is those ligand-gated potassium channels opening, which causes potassium to outflow or flow out of the cell, making it more negative, so causing that repolarization. You can also think of the ligand-gated chloride channels opening, which we haven't learned about those yet, but we will. But when ligand-gated chloride channels open, chloride will flow in. Chloride is a negative ion, which will cause the cell again to become more negative and repolarize. And that would be one of those inhibitory postsynaptic potentials. Whenever we are talking about action potentials, we see these occur at the axon and the axon hillock. When we look at that saltatory conduction, which again is that signal transduction of jumping between those nodes of Ranvier, that is occurring at the axon, where those myelin, in between those myelins and those gaps. Continuous conduction, trick question. We do not see these anywhere on our picture above because these only occur in unmyelinated axons, so axons that don't have that myelin sheath. And then exocytosis, that occurs at that axon terminal when those calcium ions flow in through those voltage gated calcium channels. So definitely work through these a few times. I would try and draw this out on your own because this is kind of a lot of information, but I've worked it out 3 times now and it makes a lot of sense to me. At first I was super confused, but after working this out a few times, it began to make more sense. So don't just do this once, you need to practice with this. All right, awesome. So moving on to number 2, this one's a lot easier, so don't worry about this. I'm gonna add some more space right in between here. So number 2, we're just gonna be using some simple calculations here. So what we're doing is we're using the Ernst equation to calculate membrane potential for each of our ions. So we'll begin with potassium. And what we see is we will look at equilibrium potassium. So this would be like if the cell membrane was only permeable to potassium, what that voltage would look like if it was only permeable to potassium. So the equation we use is 61 times the log function on your calculator divided by ions coming in, which swap means going out. Cell means in. I don't know why my iPad's moving my pen like that. So in over out. So I'm so sorry. Out over in. I can't talk right tonight. So we're going to have out over in. And once you solve that with your calculator, what we will get is negative 90.10 millivolts. When we do the same equation for our sodium ions, if our cell was only permeable to sodium, it would look like this. If our cell was only permeable to sodium, it would be at positive 65.03 millivolts. And then lastly, looking at chloride, we take that equation, 61 times log of ions coming out over ions coming in, and this will give us 0 millivolts. So with this, we can look at the word problem, and what it is asking us is when the cell is at rest, which ion would it be most permeable to? So a cell at rest is at negative 70 millivolts. Remember, we saw that up here. You just need to memorize that. A cell at rest is at negative 70 millivolts. So the one it would be most permeable to would be our potassium ion because that is the closest. So at rest, the cell would be most permeable to potassium ions. Let's say our cell depolarized, that would mean it would depolarize up to like positive 30 or positive 40 millivolts. And in that case, it would be most permeable to sodium. So just kind of work out some different examples in your head at what our cell would be most permeable to, depending on those different types of stages it enters. All right, great. And we are not going to go over this GHK equation. We are just using the Ernest equation. So lastly, before we fill out this chart, I just wanted to put our different brain areas functions into like 1 or 2 words, because I think these long definitions are pretty hard to memorize, but if we have a general overview of what each area does, it will help you remember it easier. So our hypothalamus is responsible for regulating homeostasis. So all of those, kind of things that keep us alive, like thirst, hunger, body temperature, etc. So we also see endocrine functions that we learned about last week coming from the hypothalamus, but that neuro functions is going to be working with homeostasis. Our thalamus, we can call it our sensory conductor. And I'm going to put conductor into quotation marks because what I'm talking about here is our thalamus is getting information from our 4 senses. We have 5 senses total. The thalamus isn't dealing with smell, but our other 4 senses, sight, balance, visual, seeing, and what am I missing? Sight, not smell, visual. I'm so brain dead right now. Sorry, guys. There's 4 senses. I'm not thinking of the other one. It would be a really easy one to think of. I'm just brain dead. But it'll take the information from our 4 different senses and reroute that information to specific areas in our brain, so specific lobes to be integrated. So it's kind of like this relay center. The hippocampus deals with long-term memory. Long-term memory. So for example, it kind of consolidates our short-term memories into long-term memories. So without that hippocampus, after those memories leave our short-term memories, they would go away forever. I kind of think about the movie 50 First Dates with Drew Barrymore and Adam Sandler. I guess she probably had some type of injury to her hippocampus, meaning that she could have short-term memories, but once they left that short-term region and entered long-term memories, they would just go away forever. So the basal nuclei, what we see it does is motor functions, and in parentheses, I'm going to put fine because we see that our cerebellum also deals with motor functions, but on a more gross aspect. So for example, the cerebellum would deal with like larger muscle movements. So like posture, gait, walking, balance, things like that. Whereas our basal nuclei would deal with more dexterous movements, like using your fingers, typing, writing, more advanced small movements, very dexterous, fine movements. Our prefrontal cortex deals with high level reasoning. So it's kind of that distinguishable area that makes us human. It gives us the ability to process and make sense of things. We have the brainstem, which consists of the medulla and pons, and that is dealing with our autonomic nervous system. So our autonomic control, so like blood pressure, heart rate, respiratory rate, all of those automatic controls is how I kind of think about it. Those controls, if your body was on autopilot, we don't sit there and think heartbeat, 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 vessels dilate, vessels, contract, breathe in, breathe out. We don't think about that. We're on autopilot when it comes to that stuff. So our automatic nervous system is dealing with that. And then lastly, we have the limbic system, which deals with emotions and a little bit with memory, but mainly emotions. So I'm just going to kind of put a little star because I don't want you to get that confused with the hippocampus. but it has a little bit of function with memory. All right, so with that, we can fill in this chart. So what part of the central nervous system influences our muscle activity? That is going to be the basal nuclei. So looking at that fine motor functions, basal nuclei. Whenever we look at the vital reflex centers, the cardiac, vasomotor, and respiratory, what this is talking about is that brainstem, so the medulla and pons. Brainstem, sorry. Regulating that autonomic nervous system, that's going to be the brainstem as well. Let's see, but also the hypothalamus. So hypothalamus and brain stem. I don't know why my handwriting is so bad on my iPad compared to paper. I'm sorry if this is hard to read. Whenever we're looking at the editing of sensory information, which will be passed on to the cerebral cortex, the relaying of motor signals coming out of the cerebral cortex, this is going to be dealing with the hypothalamus. Our biological clock, we're going to see that is the hypothalamus as well. Homeostatic function. Memory, integration, interpretation, discrimination, localization, and language. That is going to be dealing with the cerebral cortex, or that prefrontal cortex. Same thing. Many major reflex centers, reflex, withdrawal reflex, stretch reflex, that is going to be the spinal cord. So for example, if you were being stretched by someone and they push, let's say they're stretching your hamstring and you're laying on your back and they press too hard, your spinal cord would cause you to involuntarily push back because that stretch reflex in your spinal cord would pick that up. Same thing if you like touched a really hot stove, you wouldn't think like, oh, the stove is hot, let me pull my hand off. Instead, that signal would be sent straight to the spinal cord and we'd have a reflex of pulling our arm away without actually thinking about it. Our functional system responsible for emotion, that's that limbic system. The regulation of body temperature, that's a homeostatic functions, that's the hypothalamus. Receiving and processing sensory information from our receptor pathways like perception, that's the thalamus. Detecting information from those 4 senses. Eye reflexes, pupillary reflex, blinking, things like that. That's going to be coming from our midbrain, and we did not put that on up there, but our midbrain does visual type things. Food intake, regulating water balance, that's going to be the hypothalamus, not hyper. Another homeostatic function. And then found throughout the brainstem, reticular activating system, also involved in motor activities and visceral activities. That's going to be the spinal cord. And some of these can have kind of more functions. The brain is very complex and works as a unit. Some regions have very specific functions, but some have the same functions just in a lesser way. So this is just the key answers that Dr. Ching gave me. And then lastly, controls several major endocrine functions. That's that hypothalamus. We learned about that last week. All right, awesome. So that was it for discussion number 4. We are a third of the way done. I'm trying to get back to the Zoom call. We are a third of the way done. So we have 12 discussion worksheets total. That was number 4, so over a third of the way. Congratulations, guys. I bet you feel some sort of relief for being done with exam 1. You know what to expect now. You know how to study a little better. Don't forget that one of your exams is dropped. So if you absolutely bombed exam 1, that is totally okay. You have the complete control to replace that with a better exam score and bring your grade up again. So don't worry about that. Don't beat yourself up over that. You just have to edit your study methods and do something differently for the next exam. I love giving study tips. I am big into studying. It's what I've been doing all day. That's why I'm so brain dead right now. So if you need any study tips, just hit me up and we'll figure something out. But that's all I have for y'all today. So have a great week and I will see y'all next week.